this next section of the Unit 3 lecture, we're going to cover gastrointestinal cultures and food poisoning and those organisms that can cause food poisoning and organisms that would be considered pathogens in stool cultures. Most diarrheal pathogens are usually going to be acquired through ingesting a contaminated food or beverage. And once again, we can recover viral, bacterial, and parasitic pathogens that could be involved. So we'll talk about a few of those. Just a little bit of the anatomy. Let me scoot down here for a second. So the GI tract is mainly made up of the mouth, oropharynx, esophagus, stomach, small intestine, and the large intestine. So you can kind of see the differentiation there in the image. And normal flora that you would find in the GI tract are predominantly going to be anaerobic species. Some of the host defenses, and again, these are just kind of innate features in our body that help fight off pathogens. So, of course, our normal flora, they're going to fight off pathogens by competing for nutrients. Some normal flora is also capable of producing substances that are toxic to potential pathogens. Then we've got our stomach acidity. Most pathogens that are ingested never reach the intestines because of that very acidic environment in the stomach, which is usually at a pH of less than 4, though some organisms can be resistant to stomach acids. The intestines, so peristalsis is the term used for the constant motion that pushes substances through the intestines. So this just reduces the chances for a pathogenic bacterium to adhere to the GI tract. The mucus layer, so just like in the respiratory tract, it's able to trap bacteria. And then we've got colon. In the colon, we've got IgA, which again, we also talked about that one in regard to the respiratory tract. So it's an antibody that the body produces that fights off some organisms. Host risk factors. Median infectious dose. So this is the term that refers to the number of organisms that must be ingested in order to cause disease. So even in healthy people, if a very large number of organisms are ingested, then the body may not be able to fight them off. And then conversely, some organisms only require very small numbers of organisms to cause disease. Stomach acidity. So in patients that have decreased stomach acidity, a lot of times this can be due to different medications. They might not be able to fight off pathogens as well. Antibiotics. So if a patient has recently been on a treatment regimen of antibiotics, it can alter the normal flora of the colon, so it can predispose a patient to illness. And then patient history. So if the patient has had recent travel outside of the United States, if they have recently been hiking or camping, exposure to other people who are sick, recent dietary history. So the physician might have to take some of that information into account when determining pathogen. Okay, so we're going to break this down into bacterial, viral, parasitic pathogens. So bacterial symptoms usually present earlier than in parasitic infections. We've got Salmonella typhi. So this organism causes the most severe form of enteric fever. And enteric fever is just a systemic bacterial infection characterized by diarrhea, abdominal cramps, and fever. So it causes typhoid fever. Humans are the only known host for this organism. It's spread by fecally contaminated food. Oftentimes that's eggs, dairy, or beef, or through contaminated water, handling of animals. So this pathogen causes an inflammatory response. The bacteria are able to survive in the host immune cells, so it's considered an intracellular pathogen. And some patients can develop a chronic colonization state that leads to persistent shedding of the organism. that can cause potential transmission to others. So if you don't know the story of Typhoid Mary, there is, I think it's an article, not a video, in Moodle. Read through that. It's very interesting. And then there's also a non-typhoidal version that's not as severe. Okay, Shigella sonii is the most common species of Shigella, isolated in the United States. It's one of the most easily communicable bacterial diarrheal pathogens because it takes such a low dose of the bacteria to cause disease. It's able to produce several different toxins, including Shiga toxin, which have cytotoxic and enterotoxic and neurotoxic effects. This one is a lot of times associated with egg or tuna salads, lettuce, milk. Treatment with antibiotics for this organism is not recommended because it can cause the organism to release toxins that can make the patient sicker. Okay, Campylobacter jejuni. This is the most common bacterial cause of gastroenteritis worldwide. Contaminated poultry is a lot of times the source of infection, but it can also be contaminated meat, water, unpasteurized milk. Guillain-Barré syndrome, which is a life-threatening neurological disease, and reactive arthritis are both complications of infection with Campylobacter. The gram stain is said to resemble seagull wings, and that's what this picture on the slide here is supposed to resemble. You see how it's just kind of squiggly there in the middle, so they say that looks like seagull wings. And Campylobacter grows best at 42 degrees Celsius. So usually if you are working in a lab that performs stool cultures, you will have a separate little incubator that's set aside just for Campylobacter cultures. Clostridioides difficile, so it used to be known as Clostridium difficile. It got a name change, so it's now Clostridioides, but most people just refer to it as C. diff. So this is antibiotic-associated diarrhea caused by drugs disrupting the normal flora of the colon. So when that happens, it allows for overgrowth of C. diff, and C. diff produces exotoxins A and B. So these are just toxic proteins that are produced by the bacteria and released into its environment. They can have adverse effects quite remote from the site of infection. C. diff can also produce spores. It's easily spread by the fecal-oral route. It's not routinely recovered from culture because everyone has it as normal flora in their gut. So if we recover the organism, C. difficile, on culture, that's not really a big deal. What we need to look for is we need to be testing for, by PCR methodology, for those A and B toxins that it produces. Okay, the next organism is Yersinia enterocolytica. Infections with this organism are more common during the winter months. It's linked to meat, especially pork, unpasteurized milk, other dairy products, and water. Refrigeration does not prevent growth of the organism. This one is one that is able to withstand cooler temperatures, and actually it grows best at room temperature, which is 25 degrees Celsius, and it can produce symptoms that mimic appendicitis. Vibrio cholerae, this is the causative agent of cholera, usually found in water environments, and because it is part of marine flora, it can contaminate fish and shellfish. It's known to produce what are referred to as rice water stool, and the risk to the patient with this organism is death from dehydration. So the patient produces such profuse amounts of watery stool that they can actually, if this is not caught and treated early enough, the patient can die from dehydration. On the gram stain, the organism can resemble Campylobacter because Vibrio is a curved organism. Okay, next is E. coli 0157H7. So E. coli is also one of those organisms that's a normal inhabitant of the GI tract, but there's 5 different types of E. coli that are associated with disease. 0157H7 is a strain in the group of enterohemorrhagic E. coli's and that's what EHEC stands for, enterohemorrhagic E. coli. They produce Shiga Shigella-like toxins, so these are not the Shiga toxins that are produced by Shigella, but Shiga-like toxins associated with undercooked hamburger meat and cider. Treatment with antibiotics. Again, this is one of those that can trigger toxin production. Helicobacter pylori, and sorry, I think I'm going to skip around in the PowerPoint here for just a second. Helicobacter pylori does not cause diarrhea, but it's associated with peptic ulcer disease. One of the kind of defining features of this organism is that it is rapid urease positive. And because of that, you guys may have heard of the urea breath test. So, and that's what that is testing for, is if a patient has an infection with H. pylori, they can do this this breath test that tests for that urease production. Back up here for a second, Staphylococcus aureus. When we talked about Staph aureus in Unit 1, we mentioned that it can cause food poisoning, and this is associated mainly with mayonnaise-based foods, so like your pasta, egg, and potato salads, but also with poultry and pastries. Ingestion of the toxin that this one causes, or that this one produces, causes vomiting. Bacillus cereus, so this is food poisoning that's associated mainly with boiled and fried rice, also vegetables. Ingestion of the toxin can cause vomiting or watery diarrhea. And then Clostridium botulinum is associated with improperly preserved vegetables, meats, and fish. This one's distributed worldwide and presents with neuromuscular paralysis. And ingestion of the toxin, which is the botulinum toxin. Do you guys know what else the botulinum toxin is used for? This is what is in, what's that treatment that people can get for, you use it for migraine. You can use it for migraines, but also like to keep your skin tight. Botox injections. So they're actually using strains of Clostridium botulinum to in that treatment. And then just some other bacterial causes of GI infections: Plesiomonas, Aeromonas, Edwardsiella, Listeria monocytogenes, and Clostridium perfringens. So next we're going to talk about some of the viral pathogens that are associated with GI infection. So rotavirus, this is the most common cause of diarrheal illness in children under 5 years old, but there's now a vaccine available. Enteric adenovirus can be seen in daycare and healthcare settings. A lot of times infections are seen in immunocompromised patients, especially for whatever reason, stem cell transplant recipients. Kaliki virus includes 2 genera that are associated with GI disease, so that includes norovirus and sapovirus. Both are highly contagious and transmitted by the fecal-oral route through contaminated food, especially with salads and shellfish, but also from water. Can also be from person to person, from aerosolized vomitus, or by environmental fomites. Fomites are inanimate objects that are capable of transmitting disease. And outbreaks of norovirus are reported on cruise ships. So they're, for whatever reason, associated more with cruise ship outbreaks. And then astrovirus usually cause infections in the very old or the very young. Other sources of predisposing sources are unknown, but this one is also seen worldwide. Okay, now talking about parasitic causes. So parasitic pathogens as cause for GI disease account only for a small percentage of diarrheal illnesses in the United States. They're going to have longer incubation periods than viral and bacterial pathogens, and a longer presentation of illness. It just means the patient's going to be sicker for longer. And diagnosis is based on examination of the stool or antigen detection. So some of those parasitic pathogens are Giardia lamblia. It's the most commonly identified intestinal parasite in the United States, associated with contaminated water, especially mountain streams. So this is why they say if you, are a hiker or camper, you need to filter your water. But also it can be transmitted person to person. Entamoeba histolytica can migrate to the liver and cause liver abscesses. Cryptosporidium parvum and Cryptosporidium hominis. These organisms are known to be chlorine resistant, so public swimming pools can be sources of outbreaks. Also, fecally contaminated food or water, international produce, animals, and it can be transmitted person to person as well. Cyclospora through contaminated food, water, and produce. Most cases are seen in travelers returning from endemic areas. And then Microsporidia, so this is an order of bacteria where you've got two genus that fall into this category, and they're associated with diarrhea in immunocompromised patients, particularly patients with HIV or AIDS. A collection of specimens for GI testing. So if these specimens are not processed quickly, they should be collected and transported in Carrie Blair, Amy's, or Stewart media. So all of those are appropriate collection and transport media for GI specimens. They shouldn't be refrigerated because that can kill some of the organisms that we may be able to recover. Microscopic examination of stools for white cells. So, and that is kind of just what it sounds like. So we would take a smear of the fecal specimen, do a light smear of it on a microscopic slide, let it air dry, and then we stain it with right stain. And we're just looking for white cells, which can be an indication of some type of inflammatory disease. And we also want to avoid preservatives in the transport or collection media if cultures are requested. So preservatives should be used if examination for ova and parasites intended. So if the doctor wanted an ova and parasite examination in addition to a culture, we would have to have two separate specimens. Okay, and the culture, stool culture itself. So at the very least, stool cultures should be set up on media that would be able to detect Salmonella, Shigella, Campylobacter, and the enterohemorrhagic E. coli. So we have to use selective and differential media to plate that stool specimen on for identification of bacterial pathogens in stool. So if you remember from, I think it was Unit 1, selective media contain antimicrobials or chemicals to limit the growth of normal bacterial flora so that we can enhance the growth of pathogenic bacteria. So, and then the differential aspect of the medium is what allows us to differentiate bacterial species based on colony morphology or color changes. Okay, and some of those media, which we talked about most of these previously, XLD, Hectoin, and SS. So in those, it's going to be like one of those, not all of those. So it's kind of like the same concept as your McConkey and your EMB. These XLD, Hectoin, and SS would be interchangeable. We use those for the detection of Salmonella and Shigella. Other organisms could grow on these media, but they're going to differentiate for Salmonella or Shigella, because remember, some of those have the hydrogen sulfide indicators in them as well. CIN is for isolation of Yersinia, and that incubates at room temperature. So instead of, once you inoculate that plate, instead of sticking it in the incubator, you're just going to leave it out at room temperature. The gin, broth, and selenite, so these are both broth media. They selectively enhance the growth of Salmonella and Shigella while they suppress the growth of normal bowel flora. It can also be used for the detection of those Shiga-like toxin-producing strains of E. coli, which is the O157H7. McConkey or EMB, again, that's one or the other. Your Campy auger is for the isolation of Campylobacter, and remember I mentioned that that one incubates at 42 degrees Celsius. TCBS is for isolation of Vibrio, which inhibits normal colon flora, and it differentiates based on sucrose fermentation. So Vibrio species such as cholera, which would be the pathogen that we would be looking for, show yellow colonies on TCBS because it is able to ferment the sucrose, whereas other Vibrio species do not ferment sucrose, so you'll see like blue to green colonies. And then the last one is SMAC or sorbitol McConkey. This one is used for detection of pathogenic strains of E. coli, so that's your O157H7, that do not ferment sorbitol. So we're differentiating it from the non-pathogenic strains that do ferment sorbitol. And I'll show you a picture of that in just a minute. So here is an example of a hectowin plate, and I think we've looked at this before, but you can, this kind of greenish color here is what color the plate starts out initially. And then we've got two different organisms growing on this plate. So we've got a lactose fermenter, which is the orange one here, and we've got this non-lactose fermenting colony that also produces hydrogen sulfide. So right around the black dot in the center, you can kind of see the outer edge of the greenish, clearish colony. So it's not, that organism is not fermenting the lactose in the media, so it remains the original color of the medium. So this organism here, that's kind of that clearish green with that black dot in the center, is suggestive of Salmonella. Here's your sorbitol McConkey plate. And the sugar, this is again just based on sorbitol or sugar fermentation. The sugar in the plate is sorbitol though. So we're looking for, this one helps differentiate pathogenic strains of your E. coli. So that's your O157H7 from non-pathogenic E. coli's. XLD, same concept as the hectowin plate, but the XLD plate starts out red instead of green, and the sugar in the plate is xylitol instead of lactose, but it also has the hydrogen sulfide indicator in it as well. And then your TCBS, used for isolation of Vibrio species. Here's Campylobacter, and this is a Campy plate, but you can see the colony morphology of Campylobacter is referred to, it's kind of runny. So you see how this is like a pretty well-defined colony here, but they start to kind of like spread out almost. So colonies are referred to as wet and runny, and then the Gram-stain, again, this is the one that they say has the seagull wings, looks like the seagull wings. CIN is for Yersinia. And remember, this is the one that gets left out at room temperature because the organism grows better at room temperature. And on this media, you're looking for a bullseye type pattern. So it's kind of hard to see in this picture, but you can kind of see the dark red or the dark pink spot kind of in the middle of the colony. So that's what they're calling a bullseye. That would be indicative of Yersinia. Here's a rapid detection kit test that can be used for E. coli O157H7. So if your lab performs this test, the stool sample would start out probably in like a GN broth, and then you would take the sample, you would inoculate the stool to the GN broth, let it incubate, and then you would take your sample from that GN broth and drop it into the sample well here on the little cartridge and look for your results in the window there. Okay, time for a couple of review questions. What are some of the host defenses against GI pathogens? So normal flora, stomach acid, peristalsis, remember that's the constant motion that occurs in the intestines, mucus layer, and your colon IgA. Method of choice for detection of C. difficile. PCR testing for the A and B toxins. And remember, the reason why we don't do C. difficile detection in stool culture is because that it is normal flora. So we're looking for those strains of C. difficile that produce those A and B toxins. Okay, which bacterial stool pathogen grows best at 25 degrees Celsius? That's Yersinia. So we just looked at that CIN plate that's used for detection of Yersinia. And then what is the media of choice for isolation of your Vibrio species? And it's the TCBS. So CIN, I just said, was used for Yersinia. XLD is for Salmonella and Shigella, and your sorbitol McConkey is for detection of E. coli 0157H7. Okay, and that's the end of Lecture 2 for Unit 3.